you to basically, just like you're uploading an image to Facebook, you know, and it sucks in all the data. Once you have the collections in, which are by number, as you can see here, you know, Callaway Dodson, collection number 5719, has a family name, genus name, etc., the date, you know, all the metadata about a collection. Once you have that uploaded, you can upload the images. To upload the images, what I do is I just go in and I have a folder of images. I load the folder. I'm not going to do it now because it just can take some time, but you basically load all the images into this Excel file, drag your collector name down, and then, you know, like this could be 211, this is collection 215, you know, and you fill it in, and you hit the upload button, and then you go back to doing what you were doing, and you let it upload. It works really well from Peru. Of course, it works much faster from Brit, where we have our servers based. Okay. I assume you can still hear me, right? And then something else I'm going to show you real quick in a minute is we also have Atrium manages the vegetation plot data. So I know Brian has talked to you a little bit about ecological data. And, you know, we basically learned after we actually built a remote field database that used a sync methodology to upload back and forth between the server and the field. But we got sick of it because it's like an Access database form. We are working fast, and we find that Excel is the best technology that you can possibly use. So there's that. And what happens once the data actually get into Atrium? Okay. I'm going to go to Digital Herbarium, and we're going to do. You can browse by taxonomy. I'll just open a few windows here. By collector, by project, also research site, habitat. You know, depending on what you want to look at any given time. Let's see. It's loading. So here's an example. I'm not going to go into this, but if you wanted to, you could dive down into the plant kingdom and go into all the different levels of taxonomy to get to the plant you're looking for. My most common use of it, pretty slow right now. My most common use of it is that I'm managing projects, and my projects oftentimes are based on an ecosystem, which I'm going to get to in a moment, or they're based on a field site, like a biological station, or the Los Amigos biological station in Peru. I'm going to load this again. All right. So here you see numerous projects. I'm really proud of this one here because it's the first digital herbarium of Peru. And one of the things you need to understand is that we're not basing this on an institution. So we're really trying to get people to drop the borders between institutions so that we can start putting sets of data together for a given region, like the Andes Amazon region, which is a major hotspot that we're all quite concerned about right now. I'm going to go to just one project, an Agua Hall project, okay? Before I do that, real fast, what's an Agua Hall? An Agua Hall is a wetland, a type of wetland that occurs throughout the Amazon. We focused on research in these wetlands in southeastern Peru. Here's a view of the wetland from the sky. There are millions of palms, individual palms known as Myrtia flexuosa. We don't need to get into that. You can see here, you begin to see some savannas, which are actually bogs, carnivorous plant bogs. Well, we started getting into this and realizing that these ecosystems that are spread out along this Madre de Dios River floodplain, you know, after years of people saying they didn't really matter, they're all the same, they don't have very high diversity, it's just a pattern of river meanders. What we started realizing when we got into this is that we're dealing with something here that's never been discovered. These wetlands are situated as islands, like across the landscape, so it brings up a really interesting ecosystem model. And these are just, before I show you the Atrium project, these are just some examples of, you know, forest types. So here's an aguaje palm forest, these blackwater ponds with vanilla orchids in the background. These fern galleries that stretch at 10 kilometers, you know, 3 and 4 miles long. You just walk through these fern galleries that are full of orchids and other plants. Here's another example. This is a carnivorous plant bog. Here you have a plant that was previously only known from the Andes Mountains in eastern Brazil, and here we find it to be one of the most common dominant species of these lowland swamps. Here's a plant that has only been collected 4 times in the history of science, and the last collections we made are down here in Madrid, Peru. Here's a plant that was previously only known from the Pantanal of Brazil, which is 2,500 kilometers to the east of where we're working. And then we come to find out that about half the orchids of the entire basin of the Madrid River are probably restricted to these wetlands. Okay, so what happens is we got into this. We started realizing that at the same time, these wetlands, as sedimentary basins in this part of the Amazon, are a major attraction to the gold miners. And here you see right now a picture of a gold mine taken with probably an overflight. I don't remember if this is a Windrock image. Yeah, an overflight aerial image. You can see the gold mine. You can see the gold mines here. So basically, we set out to start looking into the conservation potential of some of these wetlands. The problem is the government, as you can see here, forgot about these wetlands when they made Manu National Park the conservation concession of Los Amigos an indigenous reserve for the Maracay Indians, and the pretty much famous Parque Nacional Bawah Soneni. And so right now we're sitting on this as an ecological model of biodiversity. So what happens when we start wanting to document the flora? I'm going to open now. Let's see, close this. One second here. So what happens? You can get into this Atrium system and you can start managing collections. You can see how many images we have per collection. You can see who the collectors are. If you want, you can view, for instance, a family checklist. I'm going to go into one family here. You can view by genus checklist. Any view you have at this system, you can export a list directly into Excel. So I'm going to go into a family that's pretty easy to deal with here in Atrium to show you some examples of things. I'll use Ananasi as an example, the Pawpaw family. And this opens up the taxonomic part of Atrium. So you always know where you are. You have breadcrumbs you can go back with. And here you can go in, you can see the summary of the genera within a given family. You can go in and you can actually see the details. So here you see the details of this. If I want to change a description, for instance, oops, sorry. So I can go in online, which is what I'm pretty much working on now with my colleagues, and I can go in and I can edit, you know. So Ananasi is in the Magnoliidae or whatever, you know. I can change this. I can add a diagnosis, trees with alternate leaves, etc. And it goes on down. Most of these fields we set up years ago. We feel like they're standard in the sense that they're really seriously useful for describing plants. The interesting thing, which I don't have time to do, is if I want, I can go in, for instance, to flowers, and I can add my own custom fields to the flowers. So I could add petals and sepals and different parts of the flower. I'm not going to show you the custom fields right now, but eventually I could. Here we go. So I just saved this. I'm working online from Peru, and I just updated this for you. And this is going to make more sense here in a second, okay? I can go to the references. We have a way to go in and add references. At any level of taxonomy, you can link out to various important databases and online information systems, such as the International Plant Names Index, the Biodiversity Heritage Library, and it sends this link through whatever taxonomic level you're at. Okay? All right, so here we go. The next thing is we have the collection system, which is where I spend most of my time. I spend a lot of my time working in these collections, and what you can see now is that for this flora, for the flora of the Aguajales, species, but I'm only in one family right now, correct? So I can go here and I can look at all collections of this family. And of course it's the Ananasi from the swamps. This can take too long maybe. Alright. And just to give you an example, let's just go into one that has a nice set of images, like this one. These were images taken by one of my field team members who is my age but didn't graduate college or didn't graduate high school. Imagine that, you know. He was a fisherman when I met him. You can go in and you can see all of the data, right? The collection data. You can also, just like you're in a museum, you can view the history of identification, but I can also go in and I can make an annotation. So I don't want to get too far away from this, but I'm going to go ahead and just give the same name so I don't have to come back to it, you know. So I'm making a determination just like you would do in an herbarium. On the right side, you can see the labels, so you always know, you know, where you're working. For those of you who don't understand the botanical information flow and how this works, it's something I guess we'll have to talk more about. You can come in here and you can also see, so by the way, look, so it shows that I updated this name on the 22nd of September. I could also go in, I could look at the images. Okay, so here you have a set of images for this one collection, okay? Now I don't have in here, let's see, I wanted to give you, let's do something else here. I want to show you herbarium specimen images, so I'm going to look for something else here. So I'm doing a search for a genus in the nutmeg family. Let's do this one here. Going back to images. Makes me want eggnog. And these are just some images of one species of. So if I go in here, you know, I can come in, and this is sort of how I work. I'm going to close some windows here. I'll go in and find, you know, 15 collections of a species, and I'll go in and I'll start opening. Oops, hold on. So you can open up an image, which I did accidentally, but this is probably one of the most interesting things here. We used a Zoomify free plugin. I don't know if I think it was free. Zoomify was used first in the medical field for doctors to share liver sections and such across the country. And you can see the details. So I can go in and identify a species. And by the way, this is the species of the source of DMT of the Amazon. You know, this, the snuff the Indians shoot up, blow up their noses. Varroa sabifera. So once I'm there, you know, I can go in, I can compare online to other databases. I can really start identifying species. Okay. We also have, like, one of the things that's important is that every bit of the collection data. So if I go in and update the collection data, it saves a new version, an old version, or saves a new version. So you can see here, you can always have the previous version or the next version. This helps in terms of mistakes. Now I'm going to go in and look at all images of Varroa sabifera. Just bear with me here for a minute. It might be a little slow to load. While it's doing that, I'll load the map. So you can see the maps of the collections in the system. It's loading. Okay, here's a map. You know, one of the things we wanted to do is be able to have sort of an online atlas, so you could see how many specimens of a given species have been collected in a given grid cell of, I think, 50 kilometers. And so here you see, you know, you have the options. You guys all know Google Earth. You have the options to go to the Earth viewing mode. You also have the options in this series, or in this on these interfaces, to download directly to Google Earth. You can download KML files. You can do little things with this, you know, move it around, but I'm not going to waste time on that right now.